Today we will discuss the final experiment or final figures that has been presented in the paper where they discussed about STK11 mutated knockout in the pancreatic cancer cells leads to the migration, and those STK knockout related migrations those STK knockout mutated migrations can be reduced or attenuated by uh, Roflumilast. So we will start with the discussion. So first they have checked the survival rates of the different kind of cells, that is the cells that they are using is TB32047 cells and another control, and two knockouts of this TB32047, two different colonies knockout of this 32047. And what they have found out, there is not much statistical significant difference that they have observed between the control and the knockout STK11 knockout cells. However, when they check the migrations, they can see that migrations in the case of STK11 knockouts, both the colonies are statistically significant or reduced by amount that is statistically significant when the cells are treated with 50 micromolar Roflumilast, whereas in the case of control we do not see any change in the migrations. And this is another representation of the survivals, that there is not much difference in the survival of the different kind of this is about the pancreatic cells. This is not much difference in the survival of the pancreatic cancer cells also and their knockout, as we can see that all of them starts to reduce at the same time with the same almost same amount of Roflumilast or same concentration. Thereafter uh, when they check the effect of migrations in the pancreatic cancer cells, they also see the same results that they have seen for this control, that is TB32047 cells, that when the STK11 knockout is present, or when in the pancreatic cancer cells there is a STK11 knockout, they can see that significant reduction in the migrations compared to the case where STK11 is present in the pancreatic cancer cells. And they did the experiment for two different concentrations, and for both the concentrations they see the reductions in the migrations, whereas in the case of control they do not see any statistical difference in the migrations. And when they did the Scratchfood assay, the similar results are reflected in the Scratchfood assay also, where for this TB32047 we can see that when the Roflumilast is used, there is a percentage of this healing is there compared to the wild type case. Percentage of healing is more compared to the wild type case. And for the control also, they see that percentage of healings are almost similar to the control case, whereas when they checked it for the knockout, they can see that the difference is visible, that when the STK11 knockout is used, the migration is more, and when the Roflumilast is used for the STK11 knockout, the migration reduced. And same thing they have observed for the other samples. And when they did the same experiment on the pancreatic cancer cells at different concentration of Roflumilast, they see the similar difference in the migrations. However, for the vehicle they do not see any statistical difference. Here also the difference seems to be there, but it's not that much. And in the case of this pancreatic cancer cells, the difference doesn't seems to be there present at all. And for the vehicles also, they do not see too much difference. However, for this 50 micromolar they see slight difference. However, when they did it for this knockout, they can see that difference is quite visible even for the second clones also, and at both the concentrations. And after validating that the Roflumilast attenuated the STK11 mutated migrations, uh, or we can say that after validating that in both the pancreatic cancer cells and TB32047, the migrations that are caused in the STK11 knockout are attenuated with the help of are attenuated. And when they did this, after finding out that Roflumilast attenuated the migration, they did the same experiment on the mice, and in the mice also they can see that visible reduction in the tumor volume, both in the case of compared to the this control case. In the control case, they see that there is a tumor volume, and they do not see much difference when the Roflumilast is introduced 5 milligram per kg per day. However, for the STK11 knockout, the tumor's volume is high. However, but when this Roflumilast is used, the tumor volume is slightly low. And same is the case with the other clone. And these things they have validated, quantified these things in terms of tumor weight, where we can see that when the STK11 knockout is used, tumor weights are high, whereas when the Roflumilast is used in the STK11 knockout, the tumor weights are low, and that is statistically different, statistically reduced, statistically validated. But when they check it for the metastasis in the liver, they do not see much difference in the liver metastasis between STK11 knockout without Roflumilast and with STK11 knockout with Roflumilast. So from this they made a point that Roflumilast, although attenuated the STK11 mutated related migrations, but uh, it has less impact on the metastasis towards this liver. Might be it might be impacting the migrations that are caused to different or other organs. So this is the final figure that they have presented, and they have made their conclusion that Roflumilast STK11 mutated pancreatic cancer cells cause more migrations, and that can be attenuated by Roflumilast. So if you need to understand more about the experiments detail, you can go through the reference that has been provided in the description, and you can also refer to the supplementary figures in order to understand their uh, main figure results in a better manner. That's all for this video. Thank you for your time.